So believing that reverse transcriptase allows single-stranded RNA to become double-stranded DNA. So this is not true, because reverse transcriptase will only use a single-stranded DNA. So you start from a single-stranded RNA. Okay, then using single-stranded RNA as a template and using this enzyme reverse transcriptase. Right, reverse transcriptase is reverse of transcription. So enzyme catalyzing the reverse of transcription means instead of DNA to RNA, you from RNA to DNA. So you synthesize a single-stranded DNA. Okay, so this is single-stranded DNA. And then usually a double-stranded DNA is needed. So from single-stranded DNA, if you want to make it into double-stranded DNA, you have to use another enzyme to make its complementary strand, right? That complementary strand of DNA that will give you the double-stranded DNA when these two combine, okay? So this DNA that you synthesize, you need another enzyme. Do you remember the name of the enzyme? DNA polymerase. Using DNA as a template to synthesize the complementary strand of the DNA. So this is our final product, double-stranded DNA. Okay, so that means you need to know the use of reverse transcriptase. It's not to synthesize double-stranded DNA, but to synthesize the complementary, single-stranded complementary DNA. So I can put here single-stranded complementary DNA. cDNA. Okay, can be SS single-stranded DNA for this one. Okay, this one. Okay, next, not realizing what promoter sequences do and linking this to transcription. Okay, so that means a student did not know the function of the promoter. Remember when you learn leg-operon. Okay, leg-operon is made up of 3 sections, which is POS. I taught you to remember POS. Okay, host, right? Promoter, operator, and structural gene. So based on that one, you know that promoter is a region where RNA will bind to initiate the transcription of the gene, right? So promoter. So promoter is a region where the RNA polymerase will bind. Next, only give disadvantages when asked for ethical and social consideration and implications. So this is a very important topic that you need to actually learn up and be familiar. Okay, in this chapter, you learn a lot about the application of gene technologies in terms of genetic engineering, gene therapy, genetic fingerprinting. Okay, so therefore, all the applications will provide us with a certain product or services that we want. So when you ask for ethical and social consideration and implication of those technologies, then you have to talk about both the advantages and disadvantages. That means ethical implication means, is it right or wrong to do it? So when there's more disadvantages than advantages, then it's better not to do. Social consideration means when you carry out the process, will it benefit the human or not? Okay, so that is called social consideration. What is the benefit? What is the disadvantage? So therefore, you must give both advantages and disadvantages in order to decide whether we should proceed to use that technology or not. Okay. Failing to distinguish between genetic engineering of a plasmid and the subsequent transformation of a whole cell. So obviously, these two are 2 different steps in genetic engineering. Okay, genetic engineering of a plasmid means you add the foreign DNA into the plasmid, which act as a vector. And the subsequent transformation of whole cells means after you add your piece of DNA to the plasmid, you actually form the recombinant DNA. And you want to insert this recombinant DNA into the whole cell. So this process is called transformation. So you need to know, okay, for each step, what is the name given for the step and what does it mean? So basically, I think these are the common mistakes among students